Welcome, and thank you for joining us today. My name is Megan Fisk-Clements. I'm a FOSSE's food science advisor. My background is in food science and biochemistry. I got my PhD from UC Davis, and I've been working in startups ever since. I am here to help FOSSE with the webinars this year. At FOSSE, our focus is to understand, translate, and intervene with food allergies. And one key question our research aims to address is, why is there such a rapid increase in food allergies over the last decades? So we're going to put up an intro slide here. Thank you for joining us in our first seminar of the academic year. We hope you had a great summer. This seminar series is a really exciting one and close to my heart. We're focusing on the Project SAFE, the science of adjuvants in food and the environment. How do these chemicals impact our health, especially as it relates to food allergies? So one question, what is a food adjuvant? So we're going to be talking about that throughout the entire series, but very simply, adjuvants are substances that are known to enhance the body's immune response. So we have a great lineup of speakers this year discussing adjuvants and these chemical components in food and the environment. We've got, let's see. Yep, you can see here on the screen all the speakers we'll have, and we're particularly interested in how these chemicals, these adjuvants, could be impacting the biological pathways related to food allergies and contributing to the development of these allergic diseases. So please join us for these monthly seminars spotlighting this critical science and learning directly from the experts in the field. Today, we are thrilled to be joined by Dr. Ruslan Metsov. He's the science founder for FOSSE, and he's kicking off the seminar series, providing an introduction to Project SAFE. So in addition to being the scientific founder, Ruslan's also the Sterling Professor of Immunology at Yale University School of Medicine and a Howard Hughes Medical Institute investigator. So we'll get into his research. Thank you for joining us, Ruslan. And for folks watching at home, I want to let you know there's a Q&A box, so please feel free to put your questions in throughout Ruslan and my conversation, and we'll get to those at the end. So Ruslan, thanks for joining us.
Thank you, Megan. Pleasure to be here.
So let's jump right in. Please tell us what systems in the gut and in the body more generally help us differentiate between food and environmental chemicals that are nutritious or beneficial and those that are noxious or harmful. What do we know and not know about this quality control system?
Yes, so the question regarding our interactions with food chemicals is really largely underappreciated. And the reason for that is because of the current environment we live in, and we don't appreciate that throughout our evolutionary history, and as well as for all the animals living in natural environments, you deal with the picture on the right, where you have hundreds of potential food sources, and majority of them are not edible, and many of them are toxic, and some lethal. And you have to thread really carefully to choose what you can eat. What we see on the left is what our food selection consists of, is just choosing between things that we already know edible. And so because this is the problem that every animal and all human ancestors, of course, had to solve on a daily basis, and the cost of the mistake could be lethal, evolution invested a lot of mechanisms to ensure that animals only consume foods that are safe to consume. And so this is just to give a very, very tiny sampling of chemicals present in edible foods. So majority of chemicals present in food, in terms of their chemical diversity, they're of plant origin. They're so-called phytochemicals. Plants are masters in organic synthesis, so they can make all sorts of molecules, and some examples are shown here. And for the most part, we don't really know how these chemicals interact with human body. There are many tens of thousands of phytochemicals that are present in natural foods. And we basically don't know the vast majority of them, how they interact with our bodies. And so that the quality control system is needed to ensure that we only consume chemicals that are either beneficial or neutral, and we don't consume anything in large enough amounts if they're toxic. And so evolution invested a lot of mechanisms. Many of them are familiar. First is olfaction. So this is a sense of smell of volatile chemicals that are derived from food. If food smells normal, doesn't have any sort of warning types of odors, like of rotting flesh and things like that, that's the first filter we use, the first passage. So we use the sense of smell to decide if we can consume food, and if it smells fine, we start ingesting it. And then there is a second layer of quality control, which is based on taste. So now we detect various molecules that are carry information through taste sensory pathways and tell us whether it's sweet, bitter, has protein in it, salt, and so forth. So there are several taste senses that we have. And one of them is bitter, and the bitter is the one that gives us a warning signal that something could potentially be toxic. So once food passes through smell and taste filters, it's ingested, gets into the intestinal system, and there is another very, very complicated chemosensing apparatus, which is basically cells of the intestinal epithelium. Many of them are specialized to detect specific chemicals. Some of the chemicals are nutrients, like as shown on this slide. This is detection of nutrients, glucose, amino acids, fatty acids. And some of them have to do with detection of noxious substances, and that we understand much less about how they operate. But in all cases, they communicate then with endocrine system as well as with the brain and with the immune system, as we're finding out more recently. And once the food gets into the intestine, these are examples of such sensory cells shown here: enteroendocrine cells, staph cells, enterochromaffin cells. They detect chemicals, whether nutrients or noxious ones, translate them into signals that the rest of the body can understand, including the brain. And from there, it continues into either absorbing them for metabolism or trying to get rid of them in some way or another. And so that's how the quality control works. And in this slide, although the details here are not very important, but what it meant to indicate is that there is a very complex interplay between this chemosensing and communication with the immune system, because that is the communication that ultimately results in either tolerating food or developing allergies to food. So this is a brief survey of current perspective on food quality control.
That is great. Thank you. There's so many details here. I'm so glad you researchers in this field are going so far into it. As we mentioned earlier, we have these adjuvants, these chemicals in food and the environment. Then we believe they are impacting the immune system and how allergies are developed. What do you think is going on here? And what is known and unknown about this?
Yeah, so mostly there's a lot of unknown. There's very little that we actually understand currently with the way that these various chemicals interact with the body and with the immune system in particular. But in general, we can break it down into two big categories. So all the various chemicals coming from natural origins, from plants, fungi, or bacteria, they can have two types of toxicity. So first, I should say that of tens of thousands of chemicals, many of them, of course, don't have any biological activity. They're neutral. They're not absorbed into the intestine, and they don't have any undesired chemical reactivities. But Many of them do, and when they do, the toxicities of these chemicals, which is common for phytochemicals because plants synthesize them often in order to defend themselves from animals eating them, basically. And so the toxicity can be broken down into two major categories. One is direct toxicity, where the chemical is directly damaging cells or cellular organelles. So, for example, if it has some detergent-like activity, it can lyse cellular membrane and cell will die. So that's one type of toxicity. The other type of toxicity, perhaps more interesting, is when chemicals have specific bioactivities, meaning that the plants actually design them in order to manipulate the animal organism in some specific way. And in that case, these chemicals can interact with specific proteins in the animal body. For example, it could be some receptors in neurons, or it could be enzymes or ion channels and so on. And the consequence of that could be toxic if these chemicals are present in large enough quantities. In smaller quantities, they may have effects that could be sometimes even beneficial. Sometimes it's effects that we actually enjoy. So one example of that would be caffeine. It's one of the phytochemicals, which belongs to alkaloids. And it binds to a receptor in our body. And that's a receptor that is involved, for example, in controlling sleep. So it binds to it and blocks it. And that's what makes you perk up after you have coffee. And so the second point I want to make here is that the complexity doesn't stop with understanding recognition. There's another level of complexity here, which has to do with what's called biotransformation of chemicals, meaning that the chemicals in their original form, as we ingest them, they can undergo further modification, either by bacteria in our gut, and bacteria in our gut have their own very complicated enzymatic machinery, and they can modify many chemicals in a variety of ways, usually for their own benefit. And the second one is our own detoxification system, which is primarily operating in the liver. So this hepatic detoxification and microbiota-mediated transformation, these are two processes then can take chemicals and either make them less toxic or more toxic, depending on the chemical and depending on the enzyme, depending on what kind of bacteria you have in your gut, and depending on your genetics as well. So all of that can result in a variety of effects, either excretion of chemicals, that would be a good outcome of defense, or if it becomes more toxic, it would cause more damage. So these are very complicated types of activities. And that's what ultimately we want to understand, because it has to do with this notion of adjuvant that you mentioned earlier, Megan, which I'm happy to expand on next.
Yeah, definitely. Do you want to do a quick expansion on adjuvants before we go on to the next question?
Yes, because this is directly related to these types of bioactivities of chemicals, the big notion that we're developing and addressing now is how the phytochemicals, chemicals present in food, in general, what are the mechanisms of their beneficial or detrimental effects on the body. And when the effects are detrimental, that's what leads to a variety of adverse food reactions. And one example would be allergy. Other examples would be celiac disease or various types of food intolerances. Mm-hmm. But the point is that what is the perspective that we're really developing, because we think it will explain biological mechanisms of food allergy. The point is that as you mentioned, Megan, for adjuvants are any types of substances that promote immune response. For allergic immune response, what is promoting it has been like an open question for a long time. And what we think is that these phytochemicals in particular, and other types of environmental chemicals, can, upon recognition by different sensors in the body, or upon manipulation of specific targets in the body, in the course of evolution, the immune system evolved to detect that as a proxy of presence of something that is not agreeable with the body or the body deems to be noxious or unwanted. And that's why it's coupled with the induction of the allergic immune response, which normally would provide a protection from these chemicals, but of course can also result in unwanted hypersensitivities.
Definitely. And I think that's such a good point, is that we understand acute toxicity. I take this, food or chemical, it has like a shocking toxic effect. Okay, that's one thing. But to have this slower, chronic, low-grade exposure of these adjuvants, I think is a whole area of research that we haven't really gone much into. So I'm so glad that we're getting to shine a spotlight on this. So building on that, how does the body develop memories of chemicals? Let's say my body had a reaction to, a peanut protein long ago, but a decade later, my body hasn't forgotten about the peanut protein. I'll have a reaction again so long after the fact. How does our body create these memories?
So that is a very fascinating question in the area of research, and there are essentially two types of memories we have, not one, but two, because this is such an important live or die decision of what we can consume as food and what we shouldn't. And there are two types of memory. One has to do with this phenomenon that pretty much everyone experienced. It's called conditional flavor aversion, or sometimes conditional taste aversion. This phenomenon is when you eat something, it's often seafood, and that would contain some toxin, and the toxin will make you feel sick. So let's say you feel nauseous, you throw up after that, but you didn't have any sensor to detect that there is something wrong with this food because it smells fine, it tastes fine, but toxin is not detectable. And then what body does, it associates something that it can detect, such as flavor, with the fact that you got sick. And this association is an example of a classical Pavlovian conditioning, and that results in very effective formation of very long-lasting memories. And the next time you smell fried calamari and it makes you feel nauseous, you don't want to eat it. So this is very common phenomenon. Almost everyone has it towards some foods or drinks. Something that made you sick in the past, and that smell you cannot stand it afterwards. Yeah. And that memory is stored in the brain. Of course, there is a lot of interesting research going on in that area to understanding how brain can form these memories so effectively and keep them for so long, because this is unusual. For most memories, it's much less effective formation. And the second memory is immune memory. And this is what we think happens, a very parallel type of principle with conditional aversion of something that made you allergic. So in this case, again, something in the food that made you feel sick, but let's say you cannot associate it with specific smell or taste, and there is no other way that nervous system can know about it, but immune system can detect anything because it can use antibodies, and immune system will use these antibodies to detect something in the food that made you feel sick, and also will form this association and form immune memory. And as a result, and this is unfortunately what happens in allergic people also, that this immune memory then can persist. But what's fascinating is that this immune recognition can also feed into this behavioral aversion later on. And this is something we've described just last year, that there is this connection between immune sensing and behavior, at least the aversive behavior.
Yes, great. I want to get to questions and answers from the audience soon, but one last question for you while I still have you. People have been trying for so long to understand the chemical makeup of food and how it's sensed by the body and what the impact of that is on human health. And yet there's still so much left to know. What do you think are the main knowledge gaps and limitations in this research? And how do you think FOSSI can answer these questions?
Yeah, so I'll go back to this slide just to give a sense of chemical diversity we're dealing with. So in this particular field, I would say almost everything is a gap of knowledge currently. just because so little is known about it.
Yeah.
And what we need to understand is, at the very least, what are the general principles by which the body detects and tells the difference between neutral or beneficial chemicals on one side and toxic chemicals on the other side. The problem is that some of these chemicals are just so toxic that just presence of one individual chemical is enough to cause some harm. But many of them are toxic in some sort of conditional way. So they need to be present in particular combinations. They need to be present maybe in the context of specific microbiota or genetic makeup of the human. And some of them, the biggest question is why some of them trigger allergic responses in some of the people and not in other people. Mm. And here, the way we think about it is that the picture should be a full picture would look something like this: that we have allergic defenses which aim to protect us from noxious chemicals. So allergic reactions to food like vomiting and diarrhea, these are protective responses that are also used against toxins. And it says when these defenses are exaggerated, then we have allergic diseases. And that's the part we don't understand why in some people it gets exaggerated. But we also think that insufficient allergic defenses, which would be in this corner, and here we don't even have a clinical name for this condition, we just predict that they exist, is when you don't have enough of the defenses. Mm. This would be similar to immunodeficiencies leading to infectious diseases. And this would result in diseases that are caused by this a lack of defense from noxious chemicals. So there is a lot to understand here, and that's why at FOSSI we're really hoping to open this new direction of research, combining food chemistry with understanding of the adjuvant activities of different foods, how that translates into immune response activation, how it translates into sensing by the nervous system, and ultimately how that can be manipulated or targeted. Because if there is one good news about the food chemistry is that once we understand specific pathways, this is probably the easiest system to manipulate because it's a natural way of delivery as they normally come with food. And so there are basically almost everything is a gap of knowledge currently, but we have some good ideas about how to proceed and how to go about it. And we hope that there would be a lot of exciting discoveries in coming years.
Wonderful. Wow. There's so much to say and so much to talk about, and we've only scratched the surface. But I want to be careful of the time and get some audience questions in. Before we go to audience questions, I do want to say thank you everyone for joining us in this seminar, and I hope that you will watch the other seminars in the series too. Our next one is October 22nd at noon. We'll be having a seminar with Noah Whiteman discussing his book that addressed what you were talking about, Ruslan, early on in the talk. All these different chemicals that we have in secondary plant metabolites and in the environment. We also, there's other ways to get involved with the foods allergy group here. You can donate, host an event, follow us on social media, and get on our mailing list. So I'm going to pull up a few questions from the audience, and I'm so glad that you're here to answer them, Ruslan. So one audience member is asking, is FOSSI conducting research on particular chemicals or pesticides that we know are in foods, such as wheat or nut orchard pesticides? There are some hypotheses and ideas about celiac disease or nut allergy being associated with these pesticides. And this ties into the question of, we talked a lot about naturally occurring chemicals. What about the artificial chemicals that are in our food system and our environment?
Yes, that's a great question. And yes, there is research at FOSSI characterizing the biological activities and adjuvant effects of food additives and other types of chemicals that are artificial chemicals that are present in our foods, in our environment, food preservatives, dyes, and so forth. And indeed, research in FASI by FASI scientists uncovered recently that some of these chemicals have indeed adjuvant activities. They can promote sensitization towards allergies. And although we haven't yet specifically looked at pesticides, but that definitely, I mean, we're looking at it very broadly. So we want to study all different kinds of activities.
Definitely. Another question from the audience. For the type 2 adjuvant activity, is that true for everybody, or would only a specific immune environment or other sort of factors contribute to some people having that activity and some people not?
Yeah, so why some people have activities and others don't, that's the question that that's a big open question. And very briefly, the answer has to do with mismatch between our evolutionary history and modern environment. So allergies, of course, growing in the past few decades, and this is mostly because of some positive and some negative aspects of modern environment, such as clean water, lack of helminth infection. Those are great things, but they're unfortunately predisposed to allergy. But regarding the antibody and phytochemicals, this is actually a great question because antibodies cannot recognize chemicals directly by themselves. They always need a protein. And that's why the way that the immune system often does it is either recognizing protein that is bound to the toxic chemical or just recognizing protein that happens to be there in the same meal. And this is the guilty by association mechanism is what makes allergies sort of mistarget often. But from evolutionary perspective, that's fine because it's better safe than sorry.
Yeah, that makes
sense. That's the logic.
Definitely. And we're getting some great questions about adjuvants. So if some of these phytochemicals can act as adjuvants, do you think there might be the other side of the pendulum swing? Do you think some chemicals might be acting in a tolerogenic way?
Yeah, that's that's a great question. And presumably, yes, we think that it's very likely that that would be the case. It's conceivable that based on what we know about how immune tolerance or sensitization works, that if you, depending on which signals you target in the gut and in the immune system, you can have either outcome. So for sure, we think that there are likely to be such activities as well. And some of them could be also tolerizing in a passive way by blocking the activity of the ones that promote sensitization. And so, so there's definitely likely to be all sorts of scenarios. And that's what we really need to get some at least basic principles with examples at least related to food sources that are common culprits in allergies, like peanuts and nuts and so forth.
Definitely. Well, I could talk about this all day, but I'm going to let you go because we're coming to the end of our time. Thank you, everybody, for joining us for this seminar, and I hope we'll see you again next month on the 22nd. Thanks, everybody.
Thank you. Thank you, Megan. Thank you, everybody.